Alrighty, good morning, good afternoon. We are going to be doing module 22, and this is going to be talking specifically about our sense of vision. So we're going to get started. All right. So this is sensory and perceptual processing. I will try to go through it relatively quickly, just so the video is not too long, but feel free to pause. And I also recommend looking through your textbook as you look through the slides and watch the video. You'll definitely get a lot more details, and there's some good, interesting readings and examples. So when we talk about vision, we always have to start with the light energy, which is the light that is entering your eye. And it has certain characteristics, and it has to do with wavelengths. How many waves, what's the height of the waves, and those characteristics can help us be able to perceive what we see. And the light energy, that's actually visible to humans, is actually only the small little spectrum of visible light. And it's about 400 nanometers, the wavelengths, and then to a little more than 700 nanometers. So anything that's below the visible spectrum, we call ultraviolet rays. So for example, bees see ultraviolet rays, and then above is infrared rays. Right? So that's the visible spectrum. So as I mentioned, we have characteristics of light, and it can give us some information about the color or the brightness of something. So the frequency, the wavelength, like how many peaks, if there's higher frequency, you're going to have more peaks here. We call it short wavelength, and those are going to be bluish type colors. Long wavelengths are going to be lower frequency. So there's less peaks. We would say reddish colors, right? So here we go. We got something blue. It's going to higher frequency than something red. All right. The amplitude is the height, and that's going to be how bright a color is. And the greater the amplitude, the higher the amplitude, the more the height, the higher the wave, that we know that that's going to be bright, versus small amplitude would be dull colors. So the hue is the name for the actual color that you're seeing. So when you say something is blue, that is the hue. The amplitude, like I said, is another name for the brightness. Your best friend decides to paint a room an extremely bright electric blue. Which of the following best fits the physical properties of the color's light waves? Well, if you know that's blue, that's going to be higher frequency. So that means it's going to be short wavelengths. There's more peaks. We also know that it is going to be having large amplitude because it's a bright color, right? So the best answer, I think, would be B, right? I've been wrong before, but I think it's B. All right, so we're going to talk about the structures and the functions of those different structures in the eye. I am going to go through them quickly because we're going to be doing more activities in class. So this is a quick introduction. The cornea is the protective covering. It's out here. All right. So luckily we have that protective covering. Just think about it as something is going to hit your eye, that you at least have that cornea to help protect. So not, all different parts of your eye are damaged. All right. And it covers the pupil and the iris, and light enters through the cornea first. Then the pupil is actually just an opening. It's an adjustable opening. It adjusts based on, the actual environment that you're in. So if you're in a well-lit area, it's actually not going to open as much. It doesn't need as much light. It's going to try to let in more light when it's actually dark and you're in a dark room. It's going to try to. So it's going to be adjusting. The iris is the portion of the eye that actually controls the size of the pupil. So it expands and contracts depending upon how much light it lets in. And also the other thing I should remember to tell you, the iris is actually the color part of your eye. So it's a muscle. And so I've got a green iris that expands and contracts, and whatever the color of your eye is. The lens is, just like a lens on a camera, it's a transparent structure that is going to change shape to try to focus on the object, on the retina, which is actually the receptor part of the eye that we're going to talk about. All right, so when it changes the curvature and the thickness, we actually call that whole process called accommodation. It's accommodating, and that the word kind of makes sense. And when we think of people that has some visual impairment, vision impairment, we most often think of people that are nearsighted, and what's happening, that's called myopia. And that is where the image is focusing in front of the retina. And then if you have hyperopia, which is farsightedness, which I think is not as common, that you are the lens is focusing light past the retina. So you're able to see far. That's why it's called farsightedness, having trouble more with things up close, or vice versa if it's myopia. All right. The retina is the most important part of the eye, I believe. That is where the rods and cones are located. And those are the receptors to help us actually interpret what we're seeing. It's where the neurons are going to actually begin the processing of that visual information, the retina, which is back here. All right. So we have the lens here at the pupil where it comes in. All right. It's going to focus with the retina. With the lens. All right. We're going to go through these different structures in class. So I'm going to go past that and continue on. The retina, like I said, it's contains the rods and cones, and it's going to receive the incoming light waves. All right, so here we are. Light hits the back of the eye. All right, so there is a little bit of a neural impulse process, chemical process. So it hits the rods and cones, within the retina, remember, and that is going to activate the bipolar cells. And I always like to remember it, the B cells, bipolar cells, which thus activates the ganglion cells, which is the ganglion cells are actually the axons of them. They all form together to create the optic nerve. All right. And the rods, there are two types of receptors, rods and cones. The rods are the photoreceptors for black, white, gray, and sensitive to movement. They're the ones that are going to be activated at night. Like if you're going to get a snack at night and you can't see, it is going to activate your rods. Your cones are going to be not responsive. And also for peripheral vision, and they're sensitive to movement. And let's see what else. They have no hotline to the brain. This is important. They share connections to a single bipolar cell and sending a combined message to the brain. Rods are sensitive to faint light and peripheral motion. So you know how you sometimes feel something kind of to the side? That would be like activating your rods. Cones are retinal photoreceptors that are near the center of the retina, and they're for daylight, well-lit conditions, which I'm, of course, today I waited till the sun fully came up to do this video. Thinking of all the details, here I am, this is one of my favorite puzzles, but all the details, this is actually a horror novel, so you can see all the details, different colors, and pictures within, the puzzle, and then when you're actually working on the details of the puzzle. So remember, I like to think also of a cone on my head. I just think of a big orange cone, like traffic cone, and it just helps me remember that cones are for color vision and well-lit daylight conditions. All right. And they're clustered in the fovea. We'll talk a little bit more about that in a second. They're not responsive in dim lit environments. And they do have a hotline to the brain. And the cone transmits its message to a single bipolar cell, which relays the message to the visual cortex. All right. So, I think that that's something new that they kind of have us really focusing on. So just remember, it does have the hotline to the brain and the rods do not. All right. We're going to do some activities in class. I'm just going to kind of go through the important things with you right now. The fovea, as I mentioned, it is clustered with cones. It's got lots of cones. And this is the part that gives you visual acuity. So when you think of when you go to the eye doctor, all right, and they have you look at different letters and you have to stand at a certain distance and you see how many lines of the letters, sequence of letters that you can read. And then they tell you what your visual acuity is. All right, so we know that that's really talking about the fovea. And, that is going to determine your visual acuity. All right. And then, of course, I guess we consider when it comes to, human vision, 20/20 is the optimal vision. The optic nerve is the axons of the ganglion cells. Remember that. So back here, this is the nerve that's going to go to the brain, the thalamus, and then the occipital lobe, which has the visual cortex in there. All right, the optic disc is at which the optic nerve leaves the eye. We actually call this a blind spot. There's actually no rods and cones there. And if you activate the blind spot, it actually things like disappear. And we are going to do a little activity of that in class as well, looking at our blind spots. So let's look at this. If you scratch your eye, which structure are you most likely to damage? A little quick review. That is the fovea. Yep. All right, so when it exits the eye, it's going to carry the impulse, like I said, to the thalamus. There's always going to be parts of the brain involved, and then the visual cortex of the occipital lobe. We will do more of these in class. So make sure you know the parts and functions. We may even have you do some sketches in class, but I recommend as you're reviewing, just start, creating a little sketch of the eye and try to figure out where the different parts are and remembering what their functions are. So how do we see color? That is another big component to vision, is seeing color. And, is the tomato red? Or is the red housed in the tomato, or is it actually in our mind, just our mind and the visual processing center that allows us to perceive color? So let's look at a couple theories. The first theory is called the Young-Helmholtz trichromatic theory. I remember it, three. I know it has something to do with three, and it's actually three colors: red, green, and blue. All right, so those are the three different types of color receptors that when you stimulate them, it can create any combination of color, and the combination of it can produce any perception of color. And I want to give a little bit more of an explanation and detail with that. So what's happening is when you take colors of light, so if I had a red flashlight and I had a green flashlight, and I wish I did, and I was to put them together, the lights were shining down, we call that additive color. It's adding wavelengths of light. It's adding more light. So when it's adding light, when you put red and green light together, light, additive color, it creates yellow. You would actually see yellow on the ground because it's adding more wavelengths of light, right? Now, often we get confused because when we think of color and adding colors together, we think of just simply adding like paint colors. Paint colors are what we call subtractive color. They pull out light. They pull out the light. So when you mix red and green paint together, it's pulling out the light. So it's actually going to create like kind of a muddy brownish color. Right? So just don't get those confused. There's additive color, which is colors of light, which is what this trichromatic theory is referencing. And then there's, when we're talking about subtractive color, that's actually paint colors mixing together. So when we think of trichromatic, often we're thinking about like people that actually have some type of color blindness. It tends to be with red and green receptors. And that is, when we're talking about the red and green colors combining together and perceiving different colors from that. And here's an example of a Buffalo Bills versus New York Jets football game. And if somebody that has red and green color blindness, that this is how the one on the left would be how they would actually see that game, which seems kind of muddy, right? That they are not having the activation of those, they're having trouble with the activation of those receptors. But those of us, that do not have it, we would see it, as the red team and the green team. All right, now you might have a favorite between those. Here are some color blindness interesting facts. 1 person in 50 is colorblind. Males are more genetically affected. It's you're not actually blind to all colors. It's simply red and green sensitive cones, or sometimes both. And vision is monochromatic or dichromatic. So we do tend to sometimes see on the AP exam things about color blindness and just those little details we were talking about. The theory that cone photoreceptors are paired together. So there is another theory, and they both kind of together give us an overall understanding of color. But this other theory is called the opponent process theory. And it says that photoreceptors are paired together, and you need to kind of remember these opposing pairs: red and green, blue and yellow, and white and black. And when you activate one of the colors, it inhibits the activation of the other receptor, all right? So when you activate the red, it inhibits the activation of the other, all right? So you're looking at something red, but it's actually inhibiting the green. A lot of times we look at things together, like different color combinations. So it's kind of doing it so quickly you don't notice what happens. We're going to be doing a demonstration in class to show the opponent process where we actually activate one color, inhibit the other ones. And then there is something, when you look at it, it creates a after image, and the after image is eventually, if you're looking at something red, I'm going to show you that example in class. All right, so feel free to look at it if you want, but when you look at something red, you eventually, when you look away, it's going to create the after image, which is the opposing color. And we're going to go into more detail in class. All right, so just remember there's two theories. There's the trichromatic theory, which is going to show that we have three color receptors, red, green, and blue, and any of those colors of light will help us perceive, the different colors. And then cones also are paired together. We have opponent process cells, and just remember those different pairs: red, green, blue, yellow, and black and white. All right. And we also recognize that our brain absolutely is involved. What we see, the colors and the shapes and the forms, it's very complex. And it actually activates different parts of the brain. And we just call those feature detectors. What are the features of what you're actually looking at? So they are nerve cells in the visual cortex of the occipital lobe that respond to the scene, whatever you're looking at, the edges, lines, angles, and movements. So I'm going to do a quick little review for a second, because sometimes we get confused, like, why do we need both theories? The trichromatic theory is going to explain how the cone receptors detect different wavelengths of light, how they actually detect the wavelengths. The opponent process is helping explain how the cones connect to the nerve cells that determine how we perceive a color in the brain. All right, going back to feature detectors. They receive the information from the ganglion cells from the retina, and they pass it to other cortical areas, and we call them supercell clusters. So it's just kind of a more detailed way to explain the complexity of what you're looking at. I mean, there's so much to, even looking at this picture of the box, there's shape, there's form, there's color, there's detail, fine details. So an example here, we got Abby Wambach, I should know her name, shouldn't I? That she had her feature detector help her be able to recognize the color, the form, the detail, the lines, the edges, so she could actually score the goal and get past the goalkeeper. Parallel processing is another thing that we need to recognize. We talk about all these different things that are happening, but it's all happening at the same time. It's happening simultaneously. You are able to look at the motion, the form, the depth, and the color of a bird flying in the sky. Okay. We actually have seagulls that are coming to visit our lake now. I guess they're flying south from being at the ocean. And just watching them, there's so much to it. It's complex. and that is parallel processing. We're able to process it all at the same time. All right, so recognizing a face, the brain integrates information projected by your retinas to several visual cortex areas, compares it to the stored information. So, I mean, just something as simple as recognizing your grandmother. There is going to be very many visual cortex. The visual cortex is going to be involved with other cortical areas to help you recognize. Some supercells, actually nicknamed grandmother cells, do appear to respond to very selectively to one or two faces in 100. Interesting. Which perceptual process explains why you can see varied aspects of your favorite singer's face and instantly recognize him or her? Best answer, parallel processing. You are correct. So we've talked a lot today. We've talked about, how when you see something, whether it's watching a soccer game and you're looking at all the different colors of the jerseys and what's actually happening in the game, you have all the retinal processing. You all have all the individual parts of the eye and functions of the eye, and the retina recognizing and able to transduce that information to your brain. And you have the feature detectors, different parts of the brain that are going to be involved for you to be able to recognize the complexity of what you're watching. And we're doing it all at the same time, that all of the color and the movement and the form and the depth all be interpreted at the same time. And then you are able to recognize and understand what it is that you are looking at. All right, so that's it for today. Have a wonderful, wonderful day.